where do we stand in our story? So last time we talked about the events of 1952 and 1953 and the discovery of the DNA helix. So the results were published in April in the Nature paper. And what happened thereafter is that Watson, in that summer, went to the Cold Spring Harbor meeting, which is a big meeting of molecular biologists, and told everybody what they had done. And most of the people there were really quite taken with it and could see the importance of it. But it should be said, I didn't really phrase it quite properly last time, although this, after a number of years became accepted as a big discovery, it wasn't accepted that way right away. There was a small core of people who saw it as important, but there were a lot of skeptics. Crick was trying to finish his thesis for the third time, and Rosalind Franklin had just left King's just as the announcement was made, and as the paper came out, she had moved from King's College and was at Birkbeck College. Both of these are part of the University of London, colleges within that university system. So let's start the question of what happened to these people after the big discoveries in 53. What happened in their later lives? And let's start with Rosalind Franklin. So I'm going to just turn my, okay. I'm going to leave it here in this mode, and you can either put it on gallery view or speaker view for yourself. So Rosalind had been, as we heard, somewhat unhappy at King's, especially with her kind of professional situation, although she was much involved in the work and had done superb work. But there were positive aspects about her return from Paris to England also. She had, in general, been trying to live on her academic income. Even though she had a private income, her family was fairly wealthy and she had money of her own, as they say. And she talked to an American friend that she had met in Paris. And this American friend said, Rosalind, why don't you use a little bit of that money to live a little better life? So in London, after a while, she was convinced by this American friend. She bought or rented a different flat, a nice one, had 4 big rooms in a nice building in South Kensington, which I think is a nice part of London. And Rosalind was a good cook. She had taken advantage of her time in Paris. She had dinner parties. It's said that she tried to introduce the English to olive oil, garlic, Parmesan cheese, basil, exotic things like that. So in her dinner parties, she invited a lot of friends. I'm gonna be reading you some things from these books today. So yeah. So it said, those lucky enough to be invited for a meal met the other Rosalind, not the one from the lab where she was, quote, someone who kept to herself and who never smiled, but rather the amusing conversationalist and attentive hostess, not so much Jekyll and Hyde. To an extraordinary degree, Rosalind compartmentalized parts of herself. Those who saw one side rarely saw the other. So quite interesting aspect of her personality. So she was working in a research institute that was at Birkbeck College, and this had been started by a very eminent scientist, J.D. Bernal, who at this point was 52 years old. He was a crystallographer, much interested in viruses, and he had gotten the financial support to start this institute at this college. So Bernal's passions were said to be crystallography, women, and the Soviet Union. He was a real card-carrying communist, a real lefty, all right. But he really wanted to start an institute that would study the structure using X-ray crystallography on viruses, and he knew that Rosalind was a superb crystallographer, and that's why he was very happy when she wanted to move to his college. So she was given a 3-year contract. Yet another little ding at her was that the salary was supposed to be 1,100 pounds, but they reduced it by 20 pounds to 1,080 pounds. Why did they do this sort of thing? I don't know. What the 20 pounds have made a difference? Just a little insult. So as she went there at first, she wrote up her work on DNA, finished that, wrote 3 excellent papers. And she sent these papers to Crick. Watson had left back to the US, but Crick was around and nearby, and Crick made very good, positive comments. And this started a really good relationship between the two of them. That summer, she took a long vacation. She loved foreign travel. And for the first time in her life, she went to Israel, which she generally liked, but she was rather upset by observing life in the kibbutz. What she really didn't like about it was the effect on the children. She said, if you choose this life for yourself, that's one thing, but to impose it upon your children so that they will have no independence or ways of making their own decisions, she did not like that. Rosalind was all for independence. So coming back to Birkbeck, she decided that she was going to work on viruses, which was the focus of the Institute, and she was going to work on tobacco mosaic virus. This is the same one that Watson had worked on for part of his time in Cambridge. And she decided on it for the same reason that Watson did. It was the model system that was chosen by a lot of labs. It was able to get the virus, able to work with it rather easily. And she was going to try to find out more details of the structure of this virus. It's made out of protein and RNA. What happened was early in 54, so she was only, this is just a year past the double helix, she got an invitation to go to a Gordon conference in New Hampshire. And the invitation was for the Gordon Conference on the chemistry of a coal. That had been her earlier work, and she was recognized as a real expert in the structure of coal. Now, let me tell you a little bit about the Gordon conferences. I don't know if anybody in here has ever had a reason to be to a Gordon conference. They're quite famous in the biological world, in the science world generally. They started in the 30s. They're held in New Hampshire because New Hampshire has all these small, private, in some cases rather elite prep schools. And the way the prep schools make some money is in the summer, they rent out their facilities, including these very Spartan dormitories for these high school age students, so that scientists from around the world can come, usually in groups of about 100 to 120. And they're rather focused on different groups. So Rusty and I went to a Gordon conference on bioenergetics for 20 years. And it's not particularly prestigious, but still, if you get invited, and they're sought after because it's usually the principal investigators who come, it's a notice that you're a player in this field. And so this was important to Rosalind, that at this international gathering, she was invited to come. Plus, she was very curious about the United States. She was a traveler. All right. So the problem was money, of course. They gave her a little bit of money, but not really enough. And then she wanted to make all of these trips around the US. So Watson and Crick both actually helped her quite a bit. They arranged for invitations. She ended up really spending most of her time in people's homes and in universities she would visit. And so she was off for her first trip to the US. What would her impressions be? So first of all, the flight itself, this is 1954. She went from London to Prestwick in Scotland, to Reykjavik in Iceland, to Gander in Newfoundland, to Nova Scotia, to Boston, finally, 11 hours late. This was not a pleasant thing to do in 1954. So we really get her impressions of this whole trip because she wrote long letters to her parents to describe what she saw. So among her first impressions here, let me read you this. Okay. All right. So the overwhelming impression, this is Rosalind's words, the overwhelming impression so far is of overabundance of everything and the resulting complete self-confidence of individuals. Superficially, they have everything and too much of it. Meals are enormous and consist mainly of protein and ice. I'm beginning to understand why they serve sweet pickles, maple syrup, raw onions, cranberry jelly, stuffed olives with all the main dishes. It's because at some stage there is a process for extracting all the taste from the main dish. But the general effect with the added spices is quite pleasant. Okay, so initial opinions somewhat mixed. But then she went on an extensive tour. I think she was here for at least 2 months, at least. She said she gave lectures at 6 different places on her coal topic. And there she found the people polite and industrial, and it was interesting. It was okay. But she really wanted to visit the science labs, and especially the virology labs. So she went to the universities in Boston. She visited New York, Madison, Wisconsin, St. Louis, Missouri. And then she started heading west. She visited the Grand Canyon and took a trip down to the bottom of the Grand Canyon. And the more and more she moved west, the more and more enthralled with the country she became. So let me read this part here. Who wouldn't? So traveling out west. Okay. So she was never shy of generalizations, but she classified Americans for her parents in this way. She said, they were at best kind and well-meaning, but incredibly ignorant and self-satisfied. On the other hand, the university scientists are as fine a crowd as I've met anywhere. They throw themselves into their work with a wholeheartedness that is rarely found in England and get real pleasure out of it. They seem very good at working together happily and unselfishly in groups in a way that is rare in England. Today, I complete the transcontinental journey. The weather is heavenly with a wonderfully clean atmosphere. Of course, they were still burning coal in London in 1958. So the more she saw of it, the more she liked it. She went to Caltech. She loved Caltech. She went to Berkeley. She felt she wasn't treated as well as Berkeley. And so one flaw in Rosalind's judgment is she ended up liking Southern California better than Northern California. But she couldn't be perfect about everything. All right, so overall, America, she loved it. So back to London, back to Birkbeck, and her position there was as a research scientist, but she also helped with some teaching, especially lab-related things, exams, etc. But she was focused on research. And early in this first year, she met a new other research scientist who was hired, 26-year-old man named Aaron Klug. He was born in South Africa. He got his training in South Africa, a master's degree at the University of Cape Town, but very bright, and then qualified for Cambridge, came to England, got a PhD in 52, very young age, and then got this position at Birkbeck College in London. So when they talked early on, Klug was working on another topic, but he saw her X-ray crystallography pictures of viruses and thought they were so beautiful from a scientific point of view that he decided he wanted to do similar work. And they talked and they started collaborating. And this was the start of a very fruitful collaboration. So Rosalind became very good friends with Klug and his family. Klug was married with children. She spent a lot of time visiting them. In Brenda Maddox's book, she said, people who knew her thought that in a way, Rosalind really developed a real love for Klug, but not in a romantic way. But she was the partner that she was along looking for. And I should say that in the end, Klug eventually in 1982 ends up winning the Nobel Prize for crystallography and viruses and nucleic acid structures. So she had submitted a paper to Nature on her work with tobacco mosaic virus. And one of the key points she tried to make is that it was a particle of a very discrete length. And she got a negative review of this paper from an eminent virologist named Norman Peary. This Norman Peary had done early work with tobacco mosaic virus, and he had said that its length was indeterminate. It could grow in all sorts of different forms. And he was a very influential man. He was kind of a grand old man of virology at this field. He had done this early work. He was eminent enough that he had a lot of influence with the funding people. The Agricultural Research Council which was the source of Rosalind's money because she was working on plant viruses. So Peary really objected to what she had said in this paper. It contradicted one of his main findings. And he had been the source of virus for her. He had been sending tobacco mosaic virus to her to work on. He stopped sending it to her, but they started making it themselves, which they were able to do. But what happened on the other side is one of the people who had done important work at Cambridge was Jim Watson. And Watson came to her defense, and so did Crick. And her paper was published, and further work showed that she was correct and Peary was wrong. But he was a thorn in her side for the rest of her career, basically. So one of her big achievements in this structure of this virus was showing where the nucleic acid was, where the RNA was. And it helped to understand how the virus attached to the plant and injected its genes into the plant, which was kind of the big biological question to be answered. She started working on a number of other plant viruses. The funding was always difficult. When Watson came back to Cambridge for a year, he kind of helped her to oppose Peary, and kind of really he and other people kind of kept her lab alive, opposing this very influential but wrong-headed old guy who was running the Agricultural Research Council. So when we got to, after several years of work at Birkbeck College, just sort of how she was doing, this is from Maddox's books, saying that worries about money apart, Rosalind was at the top of her profession and enjoying it. Invitations poured in, her contacts were wide. She held an impromptu tea party at her flat on the occasion of a conference in town. Jacques Mering, there from Paris, her previous love from Paris, attended, another person from the University of Zagreb. So it says that also, however, at many scientific meetings, it goes without saying she was the only woman on the program. Most of the women present were wives, expected to go shopping or sightseeing during the formal sessions, and to join the men only for social gatherings. All right. So now to deal with this funding problem, though, at one point, Watson said, why don't you apply to the NIH, the National Institutes of Health? And the NIH, much to its credit and to our credit as Americans, does in fact support work at other countries. And the kind of rule is, if this is felt to be essential to the progress of science, and they just can't get money from another source, we will step in and support it. And so she thought that was a good idea. And so she wrote up a grant and she submitted it. Okay. Now, in a couple of years later, she was invited again to a second trip to the US to another Gordon conference. But this time it was not on coal, it was going to be on nucleic acids. And so, as in the first trip, she arranged for many visits and many contacts. And let me just tell you what Maddox says about this second trip. Okay. So it says that as she went around, she ended up staying people and they just saw her as very good company. So Maddox says, was this sweet, laughing, humane guest the same woman who was so aloof in the Birkbeck corridors back in London? Curt with the staff, silent at the lunch table, and belligerent with the Agricultural Research Council. Memories of Rosalind treasured by those who met her abroad are like holiday snapshots, bright and smiling, while many of those from England are dark and dour. If indeed there were indeed two Rosalyns, Americans got the sunny side. And in a way, this was kind of like her time in Paris, which she so much enjoyed. And as she always said, she really saw herself as kind of a European and tended to get along with foreigners better than she did with the English. All right, there was one interesting adventure. So, she was in Gordon Conference in New Hampshire, but again, she went across the whole country. So she went to Southern California, gave a talk at UCLA. And then a virus person there, Renato Del Becco, was quite famous himself, drove her from UCLA over to Pasadena to give a talk. And just before this, it said, look, after this, a group of us, me and two others, we're going camping. I've got all the gear. You want to come? And so after the Caltech talk, they drove around to the eastern side of the Sierras to below Mount Whitney. And they didn't go all the way to the top, but they went up to 11,800 feet. Pretty good climb from the Owens Valley up there, where they got a good overview. And they went back to the car and they built a fire and they slept in sleeping bags in the kind of in the open sky and got up the next morning and were back in the lab by 5 p.m. And in the report to the parents, Rosalind she loved it. This was her kind of thing. So she got back to college. In July of 57, she was told by the NIH that they had decided to give her a grant, a 3-year grant. And they awarded her 10,000 pounds a year for 3 years. And Birkbeck College thought this was too much, that 10,000 pounds spread over 3 years would be sufficient. She got the whole grant, but just the idea that their reaction was, well, you're getting this American money, now you're going to have a lot of money, more than the rest of us, instead of saying, yay, Rosalind. She started working with polio virus. All right. But there was a kind of a problem at the time. And that is, at the end of her American trip, she started getting abdominal pains. And just putting on her clothing, she felt that she just had an abdominal bulge. And when she went back to London, they found that indeed she had a tumor. She had a large tumor. So it was surgically removed, but obviously of concern. In 57, she was well again, and she was active. And for the next year, she was kind of up and down. And the struggle for her was to get these permanent positions for her lab. She'd come in there on this limited contract, and with the support of people who were influential, the Cambridge group, et cetera, she finally did get her permanent positions, but her condition worsened. She continued to try to work. But in April of 1958, she died of ovarian cancer, and she was 3 months short of her 38th birthday. Now, was she a known person? Well, her death was noted in a story in the New York Times in America. There was a long obituary written by her institute head, J.D. Bernal, in the Times of London. So yes, she was known as a very eminent scientist, even though she was 37 years old. She had some money, not a lot of money. She had about 3,000 pounds, and she left it all to Aaron Klug and his family. And that was very helpful to Klug because Klug was living on a very limited salary. It helped him to get a house and a kind of permanent home. So let me just give you kind of from the last page of Maddox's book here, how she sums this up. So, you know, much of the controversy has been about the Nobel Prize. And Maddox says, Rosalind Franklin did not have her eyes on the prize, nor did she worry about having been outrun in a race that no one but Watson and Crick knew was a race. She died proud of her world reputation, both in coal studies and in virus research. And of her list of and she was also proud of her list of published papers that would do credit to any scientific career, let alone one that ended at the age of 37. So she has broad recognition today, and there are buildings and research centers named after her. And I just want to show you just a few pictures here. And I'm going to come back a little bit more to Rosalind at the very end. But let me. Okay, so there's Rosalind, not too many years before her death. So this is Rosalind here at a conference. This is Aaron Klug, her collaborator and very close friend. This is Crick over here. This is Crick's wife here. Okay, and this would be a typical conference picture, all these eminent guys and Rosalind kind of as the lone woman. But today, where is this? This is Chicago. And so there's a private medical school there that recently changed the name of the entire institute. And it is now Rosalind Franklin University of Science and Medicine. And there's also a building back at King's College. They renamed it for Franklin and for Maurice Wilkins. Okay. So story one of our three-part stories. Questions or comments about Rosalind and her really interesting life? Okay. All right. I think I'm going to move on then because oh, Roger, you had something you were going to say.
Well, I'm just thinking that after the Watson Crick paper, it sounds like Rosalind Franklin did not follow up in ways that one might think she could have, that she could have done more. I wonder if she could have done more with the structure of DNA to corroborate or confirm what had been done, but she seems to have left it. Is that correct? And did she appreciate the significance of the discovery at the time?
I would say, as far as the latter question, yet significant, yes and no. You know, in a way, she was really very much a chemist, and so I think kind of less interested in the biological implications. But as to not following up, you know, really from kind of 6 months before the discovery, she wanted out of King's. And she wanted an independent program of her own. And the DNA had always been given to her as something she was supposed to do. So she did not want to follow it up. And part of the deal leaving King's was, I'm leaving DNA here. I'm going to do it on my own. So other people did, and it all corroborated Watson and Crick's model very closely. Oh, okay. Ron, I think I'm going to move on because I'm really kind of tight for time today. It's just so much interesting stuff. So let's move on to Francis Crick. So before we go into Crick and Watson, both of whom did follow up on DNA, I think it will be best to give a sense of what we know now, to kind of put it into context, because it's sort of difficult to, especially, if you don't really know a lot of biology, to understand the significance of what they were doing without knowing an overview of what we're talking about. So what we know now is what's called the central dogma of molecular biology. DNA makes RNA makes protein. So we know that DNA is a code, and it is copied. The DNA is copied into RNA, and RNA is a molecule that in its chemical structure is almost exactly like DNA with just very tiny differences. And then this RNA code is then somehow used by a machine, the ribosome, to make proteins. And I'm just going to show you two more slides here. So overall, what happens is that the DNA, which is this double helix, has an enzyme that can sit on it and pull the two strands apart. And then one of these two strands is copied into an RNA molecule. So the RNA has a sugar phosphate backbone with the 4 nucleotides copied from the DNA. Only difference is that in DNA, it has a nucleotide T, and in RNA, there's a subtle chemical difference that it has the nucleotide U. It's thymine versus uracil. The RNA then leaves the nucleus, and out in the rest of the cell, it binds a very large structure called the ribosome. I'm going to show you a picture of the ribosome next. And the ribosome is the actual protein synthesizing machinery. So the RNA is kind of read as a tape going through the ribosome. And the code on the RNA is interpreted, if you will, by this intermediate molecule, the tRNA. The tRNA says CGA carries a particular one of the 20 amino acids in protein. And so this carries in and binds to the RNA, holding on an amino acid, and then the ribosome puts them all together to make a new protein. Now that's a kind of quickie overview, and it's a complicated process, but I think it'll help you to understand what Watson and Crick were doing in the later part of their life. Now, lastly, let just me show you that the ribosome, the molecular machine that does this, turns out to be a very complex, huge molecular machine. It's 10 times bigger than a typical protein or enzyme. And it's also very different from the other kind of molecular machines in the cell in that it is made mostly not of protein, but actually of RNA. So in this picture, what you have here is the thin segments, the kind of purpley ones. These are proteins. There are about 50 of them, 50 little proteins. And oh, somebody else needs to mute themselves there. And they are bound to,
Now I gotta get myself.
I'll go back and do that. Bound to the gray part, which is RNA molecules. And then the other part of the ribosome here, the kind of light colored ones here are the RNA, and the purpley ones are proteins. So a huge complex molecule. Okay, so let me go back here. All right, so with that context, that's what needs to be discovered. What was the role of Watson and Crick in doing this? So did Crick ever finish his PhD? Well, the answer is he finished it 2 months after they published the double helix. You know, it was time to wrap this up. And he ended up using all of his work on proteins. So he actually did do his separate project, and is the structure of hemoglobin, the oxygen carrying molecule on our blood. And his thesis had useful data, but he had made no big breakthroughs. It was very difficult to solve that structure with the techniques they had available at the time. Then he had arranged for a year postdoc in the United States who's going to go to the Brooklyn Polytechnic and really kind of continue some protein work. He found the work in Brooklyn to be dull and depressing. His wife, Odalie, was not happy. They were in a very drab apartment in Brooklyn somewhere. The only high point was the summer conferences. He got back together with Watson, and they were talking DNA and how important that was, and convincing people, and other people could see that this was the route to understanding what genes really did and how they operated. They also thought more and more about it and thought that it was maybe wasn't the DNA itself, that maybe it was the RNA that they knew was somehow copied from the DNA. That was important. And so there were a group of them at one of these conferences said, well, let's form a little club. We'll keep in constant communication about where we stand. There are 20 amino acids, and we'll allow 20 members. And it was called the RNA Tie Club. They had a little tie clip, and if you belonged to this club, you became one of the RNA Tie Club people. And this was a very important group in the development of molecular biology. So Crick goes back to Cambridge. He had gotten a contract for 7 years. The importance of what they had done was recognized by the Cambridge people. And he seemed, what was he to do at Cambridge? As far as we can tell, it was pretty empty, open, just think brilliant thoughts and see what he could come up. But he struggled because Crick operated by conversations. I'm going to just a second here. I'm going to mute a couple of people. Such power. All right. Okay. He didn't have Watson to talk to anymore, but his good luck, within a month of coming back, he ran into this guy. And this guy's name was Sydneybrenner. I can see a bunch of you. How many of you ever heard of Sydneybrenner? Interesting. I see 1 or 2 hands going up. Sydneybrenner was probably the most brilliant person in this whole story. And people who know molecular biology know Sydneybrenner. He did win a Nobel Prize, among other things. But anybody who has ever met him, this guy was brilliant. So Brenner, again, like Klug, was born and raised in South Africa. He went to the university at 14, a year younger than Watson did. Watson was 15. When he was 20 years old, he qualified as a medical doctor, as an MD. He got a scholarship, surprise, surprise, to go to Oxford. And he was trained in bacterial genetics. So unlike a lot of these other people who came from a background in physics, Brenner was a real biologist. He really knew biologists from the beginning, although he was interested in everything. And he had read about this mathematician von Neumann's work on the early days of computing and in codes. So he and Crick got to talking about the idea that DNA may be a code. Brenner was very enthusiastic about it. He got Crick to be enthusiastic about it. Because, at the time, it seems odd to us now, but if you put it in the complex of the time, the molecular biologists thought, well, we got DNA, it seems to be the gene. How does it do it? Well, it must fold up like some giant enzyme, and then amino acids will stick to it, and it'll put the amino acids together to make protein. And Crick and Brenner talking, so, well, maybe it's not that direct. Maybe it just has the information, but something else actually makes the protein, which turns out to be correct. All right. So one of the things that Crick did his whole career is he read just enormously. I mean, people said that Crick could sit down and just nonstop read for 2 hours. He could read for 8 hours just straight. And Brenner would say, "You're reading the dull papers." And Crick would say, "There might be a clue in it." So this was his career. Conversation and reading was how Crick did biology. So that summer, they heard that there'd been a big breakthrough by the biochemists that somebody had figured out how to make DNA in a test tube, independent of a cell. They could break cells open, extract just a few of the components. They didn't know exactly what they were, add radioactive amino acids to them, and get a radioactive protein. You can't kind of overstate the importance of the availability of radioactive molecules that occurred post-World War II. When we had the first nuclear reactors, you could suddenly make radioactive phosphate, radioactive carbon, radioactive calcium, almost anything you wanted. Because what you now had that you never had before was the ability to put a label on atoms and put them in a mixture, and you could follow them because they were radioactive. You had a Geiger counter, you knew where they were, and you could find out what happened to them. So this played a role in so many important aspects of biology. It was key to understanding how photosynthesis works, one of the most important biological reactions in the world, probably the most important. All right. So anyway, they'd figured out how to make it. They found out that the key thing in the cell was something they called microsomes. This turned out to be ribosomes, but they didn't know it at the time. There was just some heavy particle in there that, with a little bit of RNA in it, somehow made protein, but they were trying to figure out how it worked. So Crick was reading all of this. And in 1958, he wrote a paper that was called On Protein Synthesis, published it in a major journal. And it really postulated the core ideas of the central dogma of molecular biology. And he, in fact, invented that term, that DNA must be a code. It's copied into RNA. The RNA code somehow goes to these microsomes, and they somehow use that information to make protein. And so he was, in a way, kind of directing the whole operation around the world, in a way, with his ideas, synthesizing ideas and papers that were coming in from labs all around the world. Now, at this point, they didn't really kind of know what happened to the RNA, and that was puzzling. So there was one really interesting episode. It was a little bit like one of the stages of discovering the DNA structure. I want to tell you, this is, I'm reading now from Matt Ridley's book about Crick. At one point, they were trying to figure out how all this worked. And some of the major players in this were in Paris. This is Jacob and Monod, who were working out how genes are turned on and off. They weren't doing any biochemistry. They did it all with genetic experiments, really ingenious things. But it was Jacob that came by, and he was telling them about these experiments that he was doing. And so they were doing their usual thing. They were having a long conversation. So let me just read you this part here. It said, in April of 1960, Jacob was in London for a conference, and he traveled to Cambridge. And the labs being closed, Crick, Brenner, and Jacob, and a few others gathered in Brenner's office in King's College to listen to Jacob talking about his experiments. Jacob found a skeptical audience. Francis and Sydney made me take a veritable examination with questions, criticisms, comments, a pack of hounds racing around me, nipping at my heels. Jacob held his ground and described his experiments. Then suddenly Brenner let out a yelp, and he began talking fast. Crick began talking back just as fast. Everybody else in the room watched in amazement. Brenner had seen the answer, and Crick had seen Brenner see the answer. The ribosome did not contain the recipe for the protein, the ribosome itself. The ribosome was just a tape reader. It could make any protein so long it was fed the right tape, and the tape was some kind of RNA. All right, so this was kind of a critical moment. They said, there's no super enzyme. It's just a piece of information that's fed into a machine. The ribosome is the machine. All right, so that was critical. And so to Crick's credit, whenever he got these ideas, he wrote them up. He described why he thought it was this way. But in this case, Crick did something rare for Crick. He said, I got to do an experiment. I'm going to have to go into the lab and get my hands dirty. But he got really enthusiastic about it because he thought he had a good experiment. So he was using bacterial viruses. And this is kind of complicated, which I won't go into, but suffice it to say that without actually looking at the molecules, they could do the genetics with sufficiently high numbers of crosses and mutations to really figure out how many changes there were in the DNA. So they found that certain chemicals would cause the DNA to lose bases or to add bases. What it was doing was it was interfering with the copying mechanism, made it inaccurate, but sometimes it'd make it too short and sometimes too long. And what they found with these experiments is that if they deleted one base or two bases or added one base or two bases, it would inactivate the gene. However, if they added three or they deleted three, then the gene would usually be okay. And Crick describes going in one evening with his technician, getting out these plates that allowed him to see the types of mutations that would work. And it just fit his three hypotheses. And he said to his technician, this evening, you and I are the only ones in the world who know that the DNA code is a 3-letter code. And he was right. All right. So in 1962, the Nobel Prize. Okay, he and Watson won the Nobel Prize. They kind of knew it was coming. At this point, this was 9 years after the discovery, all the data was showing that the model was correct. And to the biologist, it was showing deep insights into how genes functioned. So he had hints that it was coming from Monod in Paris. The Nobel Prize Committee had asked Monod for some nominations and for a kind of preliminary write-up of the work. So Crick wrote to Monod. Now, this is before the announcement, but when he knew that they were thinking of giving it to him and Crick, and this is interesting what letter said, what Crick's letter to Monod said. He said, quote, the data which really helped us, he and Watson, to obtain the structure was mainly obtained by Rosalind Franklin, who died a few years ago. Okay, but then Ridley says, but Nobel Prizes are never given posthumously, nor is a single prize ever shared by more than 3 people. Had Franklin lived, the Academy might have solved its problem by giving her and Wilkins the prize for chemistry and Watson and Crick the prize for biology. All right. As it was, the chemistry prize went to Perutz and Kendrew, who were the senior people in Cambridge working on proteins. And it made nearly a clean sweep for Cambridge. So Watson, Crick, Wilkins, Perutz, and Kendrew. All right. That was a big year for Cambridge. Okay, so he got the money. Interestingly, the money, nowadays I think the Nobel Prize is, I don't know, it's something like $1.5 million or something. They got $16,500. Not a huge amount of money, but it was enough money to allow him to buy a house. And Gunther, this is for you. He bought a half partnership in a sailboat, a big boat, 42-foot boat. And he sailed it for a year or so, but he couldn't handle it. And he finally sold it and he bought a motorboat. And he kept the boat at a place near Naples, and he would spend a significant amount of each summer going around in his boat. So Crick's big focus now was the code. We know that it's a code. So what is this code? And, you know, he was reading everything that he could find, and he kind of tried to work it out himself. He had some ingenious ideas about the code. One was just so pretty it couldn't be wrong, except it didn't fit any of the data. And so he said the data win. So in 1961, just the year before they won the Nobel Prize, there was an international biochemical conference in Moscow. A lot of people, a lot of little talks. And there was a kind of a talk by somebody nobody knew, a little 15-minute talk in a small room, but one of Crick's friends had heard this talk. It was by a guy named Michael Nirenberg, who worked in some small lab at the NIH in Washington. And this guy said, Crick, you've got to hear this talk. And he told him what was in the talk, and Crick, who was one of the big organizers, said, you've got to repeat your talk to the whole conference at the end. So what had Nirenberg done? Nirenberg was working with the fact that they could make a protein in a test tube. And he had worked out the test tube, the methods to make RNA molecules that had only one letter in them. So RNA is A, C, U, and G. He made an RNA that was all U, just U. And he found that it would make protein. The protein, though, only had one amino acid in it, and it was phenylalanine. And while he was at the conference, his graduate student called and he said, I got the poly-C to work. I made RNA that's only all C's. And he said, and it makes a protein that's only proline. And so Crick and the others saw immediately, we have the beginning. We know that the three letters U says phenylalanine amino acid. C means proline. And so you could now think of, let's make RNA in which 2/3 of it is C's and 1/3 of it is U's. And we can predict there's just a few possible combinations. And what are the abundant amino acids and what are the less abundant ones? And we can work out the codes. Now this turned out to be, you know, that worked for some of them, but then it gets harder and harder. But basically that was the breakthrough. And I remember as a graduate student talking to somebody who had gone to that conference and heard the talk, the second talk. And there were labs around the world, some really powerful hotshots at MIT and Harvard working on this coding problem. And here comes this obscure guy. And this person related this conversation. He said, one of the people who worked in this lab, he said, you know, the guy next to me said that talk was electrifying. He said, but I felt electrocuted because he had been scooped as to how the code read. But they needed to work up all the rest. I mean, after all, if you think this through, if it's a 3-letter code and you got 4 possible kinds of letters, that means that if any combination of 2 will only make 16 possibilities, but there's 20 amino acids. So you had to have 3. So that fit Crick's idea that there was a 3-letter code. But that gives you 64 possibilities to encode 20 amino acids. So how does that work? So there was a lot of work to be done. And so there was 5 years of work to be done. And Crick was the critic, the scorekeeper, the interpreter, but they finally did work it all out. And I'm just going to show you this quickly here. All right. So this was the ribosome. Okay. So when it worked out, here was the genetic code. And so this is the codon table. So the way you read this is that of the 3 letters, if the first one is a U, the second letter is a U, and the third one is a U, UUU, that will encode phenylalanine. Now, it turns out if you have UUC, that also encodes phenylalanine. Or they found out that if the code reads CUU or CUC or CUA or CUG, it doesn't matter what the third one is, it all encodes leucine. And this was something that Crick kind of hypothesized about. It's called the wobble hypothesis, that as long as you match up the first two, it can kind of wobble on the third. So with time, they also found out that 3 of the combinations did not encode an amino acid. Instead, when you got to those combinations, UAA, UAG, for example, it would stop. And so this is the termination. So it would read through the code until it got to one of these 3-letter combinations, and that would say end of protein, stop here. So it was all worked out, took a lot of work. There were a couple of Nobel Prizes that came out of this, but it finally gave you the essence of the code. Now, Crick's life during this time is he had, a few years earlier gotten a visiting appointment at the Salk Institute down in La Jolla. And so in February, he would spend a month in La Jolla. Then in July and August, he would be on his boat off Italy or Greek islands. And then he would go to summer conferences. He got involved in a number of other projects. He got very much interested in the proteins that are associated with the DNA. One thing is he was really mystified by the fact that it became clear that all organisms use the same code. And these 3 letters specify the same amino acid in bacteria and plants and prokaryotes. And that was a mystery, and it led him into the idea of it must mean that maybe we were incubated, seeded by some mysterious living creature from another planet, galaxy. It's the idea of pangenesis. And he really thought seriously about this as the only possible explanation for the universality of the genetic code. But then in 76, when he was out to the Salk offered him a permanent position. Said, why don't you stay here? We will give you a nice package. Among other things, we'll give you enough money that you can bring in anybody in the world that you want to talk to. You like to talk to people. And we'll put them up for a couple of weeks or a month or 2. And Crick at first was reluctant, but the weather was so nice and the idea was so good that he just did not go back to England. I mean, went back for vacation, but he accepted the position. And he became a member of the Salk Institute in La Jolla, in Southern California. And he decided, early in life, we may remember back from the beginning of this story, when he was a physicist, frustrated physicist, he decided he either wanted to solve the secret of life or the secret of the brain. He decided, well, I did the life thing. Now I'm going to do the brain. So he got very interested in the brain and consciousness. And he did his Crick thing was to do intensive reading, 2 to 8 hours a day, everything he could learn about it, brought in experts. He needed a Sydneybrenner or a Jim Watson, and he found one, a neurobiologist from MIT named Christoph Koch, who had moved to Caltech. And for the next 18 years, he and Koch would talk for hours every day. So he really wanted to be able to connect conscious thought with neurons in the brain. He said, let's reduce this to as simple a problem as possible so that we can be conscious of maybe 2 different things. And then let's see if we can figure out what brain part changes when we're conscious of this subject versus that subject. And he worked hard at it. It turned out to be a very difficult problem. And as we all know, he hasn't solved it. Consciousness remains probably the big unsolved question in biology. And my reading of it is we're nowhere close to understanding it. He did write a book, which he called The Astonishing Hypothesis, that made the study of consciousness a respectable area of study, but no major study, no major breakthroughs. 1994, he became the president of Salk Institute for a year, took it very seriously. It only did it for the 1 year, but did it. And then as he got kind of into his 80s, and when he was 82, he bought a big piece of land out in the desert, 100 miles east in the Borrego Desert, east of La Jolla, and built a house, got very involved in desert gardening, but he never stopped working to his last days, never stopped reading. He was active literally to his last days in the hospital. So 2001, he was diagnosed with colon cancer. He was treated, he lived for about 3 years, you know, better days and worse days, and died in 2004 at the age of either 87 or 88. So let me just read the summary that Michael Ridley has about Crick. Quite interesting. So because of the momentous nature of his discoveries, Francis Crick must eventually be bracketed with Galileo, Darwin, and Einstein as one of the great scientists of all time. Like them, he discovered a great truth that took the world by surprise. In his case, the nature of life. Like them, he made many discoveries, not just one. Like them, he invented and dominated a whole new discipline. However, he did none of these things on his own. So does he deserve to join this pantheon for his genius, or was he just in the right place at the right time? He will always be known for being the first person with Watson to see how DNA works, a completely unexpected result. But in some ways, this greatly underestimates his achievement. So it's no doubt that Crick did an enormous amount. He was one of the great thinkers. He was really kind of unusual example in biology of a theoretician who really, really accomplished major things. So let me just show you just a few pictures from Crick. So here he is doing a radio broadcast for the BBC. He's probably about 40 years old at this point, and they did have a child, and they took a lot of trips. He was described as a good father, but not the type who took you out to play. He was always reading manuscripts, even on vacations. And this is Francis Crick and Odalie in their later years. And like Franklin, he has become a famous man. And this is now the Francis Crick Institute in London. It's one of the world's huge research institutes and kind of aptly named after this person. So, James Watson. Questions, quick questions here on Crick. All right, let me move on to Watson. And maybe I give a warning here. I think I'm going to run about 10 minutes over, folks, but come on, this is really good stuff. So hang in there with me. All right. So Watson, you know, save him for last because, you know, he was the key to the whole thing. You really have to, whatever we think about Watson, he was the centerpiece. You know, it was his idea to go after DNA. It was his insistence that they solved the structure, and he became just a central player in this whole era of biology, you know, through his life. And Watson is alive today. He's 93 years old and still going today as we talk. So after 53, the model came out. He decided that DNA seems to make RNA, and let's focus on the RNA. So let's get the structure of RNA, because maybe RNA folds up, amino acids bind to it, and that's what makes protein. He spent 2 years working on this with some really good people, and they just could not get a structure. It just seemed to be a diffuse thing. And we now know that RNA doesn't fold up into a disc structure. It's just single-stranded stuff. I mean, there are discrete structures, but if you isolated RNA from the cell, you wouldn't find any particular structure. So he became a part of, one of the founding members of the RNA tie club, and was in this group. So it became clear that Watson was a sharp guy and had made a major discovery. And so he got to be an assistant professor at Harvard. That's, of course, Cambridge and Harvard, the big places to be a professor. Early on, he got the news that teaching would be an important part of this. It's interesting that Harvard has always had the possibility, what do I want to say? Anyway, a rule that we have all of these prima donnas here, but we are a teaching university, and they have strict rules that if you are a Harvard professor, you have to teach. And so Watson took his teaching very seriously. He got really involved and very much enjoyed his teaching. That was an important part of his career. He got his first grant from the National Science Foundation, and it was to study ribosomes. They knew that these microsomes, these ribosomes, were the key to it, and allow him to buy an ultracentrifuge. It was a very expensive piece of equipment at the time. I don't know. Today, they're still expensive. Although, you know, $60,000, $100,000 by a good ultracentrifuge. And it would allow you to study these particles. And what came out from the early days was the discovery that ribosomes were much more structurally complex than we anticipated. I showed you that picture. A typical ribosome has something like 70 different kinds of proteins and 3 huge RNA molecules, very complex, and its structure has only been solved in the last few years. However, they did find out that it seemed to be of a kind and very stable. It didn't vary that much. So the ribosome itself didn't seem to have the information to make these thousands of different kinds of proteins. And that was a mystery. Now, I should say that when I was trying to find out what Watson had done for his scientific work, I don't know that there's been a biography written yet of Watson. He's written a couple memoirs himself, and the one, I put it on that reading list that I sent around here. This is a memoir he wrote when he was about 70. Trying to find out what were his scientific achievements. And I looked in Wikipedia, which tends to be thorough, and they really kind of dismissed it with 2 sentences saying he really didn't do much of significance in science. And I think that's not fair. That's not fair at all. In fact, Watson did some very important things. For example, one of his most important things, in 1960, this is sort of 7 years after the double helix, he was infecting cells with a virus. He knew that the virus would make its RNA, and this RNA would then somehow take over protein synthesis. And he and his graduate students were able to show that what happened was the RNA got associated with the ribosomes in a transient way. He had discovered messenger RNA, big deal. And he writes in his memoir that he made one of his biggest mistakes in his life, and that he decided that he was going to hold back publication until he got all the details, just like Rosalind Franklin. And he said it was a big mistake because he did that key experiment in February, and just 2 months later, over in Cambridge, Brenner and Jacob and Crick were having this discussion when they discovered messenger RNA. And Brenner then went on to do the experiments to show it. And Watson got scooped by Brenner and Crick. So he said that was a big mistake. If you find something big, publish it right away, he said. But he continued to do good experiments. He had many students and postdocs who became very prominent, eminent molecular biologists. Now, the other thing, he's now a Harvard professor, and he tried, even as an assistant professor, to change Harvard. Just 2 years after, and I think his 3rd year after he was there, he got tenure, which is quite early. Now, really interesting, I've kind of in the academic community heard about this aspect of it. So one of my all-time favorite books is this from E.O. Wilson. People have maybe heard of E.O. Wilson. He's a great person who understands ants more than anybody else, but he's written a lot. So let me just read this little section. Wilson, in his memoirs, has a chapter titled The Molecular Wars. And here's what it's about. He said, when he was a young man in the 1950s and 1960s, I found James Watson to be the most unpleasant human being I had ever met. He came to Harvard as an assistant professor in '56, also the first year that I came as an assistant professor. Watson was 28. He was only a year older than I was. He arrived with a conviction that biology must be transformed into a science directed at molecules and cells, written in the language of physics and chemistry. What had gone before, traditional biology, my biology, said E.O. Wilson, was infested by stamp collectors who lacked the wit to transform their subject into modern sciences. He treated most of the other 24 members of the Department of Biology with a revolutionary, fervent disrespect. At department meetings, Watson radiated contempt in all directions. He shunned ordinary courtesy and polite conversation, evidently in the belief that they would only encourage the traditionalists to stay around. His bad manners were tolerated because of the greatness of the discovery he had made. Watson, having risen to historic fame at an early age, became the Caligula of biology. Anyway, this, by the way, is one of my all-time favorite books. Naturalist. It's E.O. Wilson's memoir. Lots of good academic gossip in this book too, by the way. And I tend to believe E.O. Wilson's side of the story here. Anyway, so Watson, to his credit, did push in bringing successful senior people in. He pushed the careers of the young molecular biologists. There are guys like Wally Gilbert and Mark Potashny who were famous in molecular biology for working out how genes are turned on and off. He pushed for new lab space. Interestingly, this really had an effect on the development of UC Santa Cruz. And the connection is this. One of the senior professors at Harvard was Kenneth Tiemann, a plant biologist who had made major discoveries in plant biology. Watson thought plant biology was worthless. And Tiemann was coming up to retirement. And so he said, I've had it. I'm going to California, where they're founding this new branch of the University of California. And so Kenneth Tiemann, kind of in disgust at what was going on in Harvard Biology Department, kind of left and came out here and was a founder in a very good way, in a very effective way, of science at UCSC. And was really, among other things, one of the people who really insisted from the beginning that we have PhD programs and research programs. All right. So anyway, 62 Nobel Prize. In Watson's book, he has a long description of what it's like to go to Stockholm. It's interesting. It illustrates one of Watson's other distractions. About a third of all of Watson's books are about looking for pretty girls. All right. And in Stockholm, he talks about how he wanted to sit next to the Swedish princess, Desiree, all right, and had some success. Richard Feynman wrote him, I have to find you this one. He talks about this. Richard Feynman wrote him a congratulatory telegram, but Feynman, being at Caltech, and Watson had spent a lot of time in Caltech, knew Watson. This was how Feynman congratulated Watson on winning the Nobel Prize. He said, and there he met the beautiful princess, and they lived happily ever after. All right, so he knew what Watson's preoccupations were. All right. So there's much in his book about trying to find girls. Now, fortunately for all of us here, I have invited an eyewitness to Watson's behavior to tell you a little bit more about this aspect of Watson's personality. So my eyewitness can come on the line here.
I'm here.
This is what Rusty.
Okay.
Why, those who don't know it, Rusty and I are married. Okay.
Okay, so what happened is after I finished at Duke, I spent a year and a half working in my uncle's lab at Harvard. My uncle's lab happened to be on the same floor as Jim Watson's lab. Anyway, so Watson had this reputation of being really interested in looking for pretty girls. Anyway, one of the women in our lab was actually a high school student, and Watson seemed quite taken with her. Now, I don't remember now the details of why I thought this, but in any event, and I have to say my uncle was one of those people that Watson would have thought needed to be dispensed with. Anyway, so when Watson won the Nobel Prize in 1962, he had a party at his house. And one of the grad students invited me to go along with him. I guess everybody in biology was going. So Stanley introduced me to Watson, and he just frowned at me and looked very unhappy that I was there. And I had to conclude that I was not a good-looking woman in Watson's opinion. So that's my story of Jim Watson.
All right. Okay, that's just one witness, so. All right. Now, so during this time.
Nobody said that he had good judgment, though, correct?
Right. Okay, so one of the things Watson did was he was a good writer. He was a good writer, and he wrote two very important books. One book was The Double Helix, his memoir, What Happened. And I'm going to come back to this at the very end. The other is that as part of his teaching, he took his lecture notes and he wrote a book called Molecular Biology of the Gene. And this is my copy of this book, was published in I think, let me see here, 1965. And I remember as a young student reading this book and say, gee, this really presents all of this material in an understandable, readable way. He's just really good at it. Good graphics, good explanation, very logical. And it became a very important book in kind of the development and study of biology. When I came to Santa Cruz, I mean, after a few years, so I came to Santa Cruz in '79, this is 10 years or so after this book came out. And sometimes some of the older professors would say, in the old days when we taught intro biology, we taught from the core material. We used Watson's Molecular Biology of the Gene instead of using one of these modern textbooks. And my answer to that is, we should still be doing that. And I actually think if you took this 1965 book and the students learned what was in this book, they would have a comprehensive view of 85% of modern biology. Just very well done. Anyway, so kind of moving on in 68, Watson's 39 years old, and two big events in his life. First of all, I mentioned in the past that Watson had gone to these conferences in Cold Spring Harbor as a graduate student. He really first met really famous scientists at Cold Spring Harbor. He continued to go there. Cold Spring Harbor is a small private research foundation on Long Island. And they asked him if he would become director. It was struggling as a small private place. It had money issues, it had staff issues, and he just had a fondness for Cold Spring Harbor, but he also had a fondness for Harvard. He was very loyal to Harvard. He'd gotten lots of very lucrative offers, including from California schools. So he decided he would become director. He worked out a deal with Harvard that he would continue his appointment there and his teaching there. And he continued at Harvard for another 8 years. But more and more, he shifted his activities to Cold Spring Harbor. He had to address the money issues, the building, the staff, and he was successful. He was able to use his celebrity to really bring in money and restore that place. In March of 1968, there was a 19-year-old Radcliffe Jr., not a scientist, who was helping his secretary. He thought she was very attractive. And basically, he got involved with her and somehow was able to convince her that he was a guy worth going with. This was had been a big problem in his life to that point. And the bottom line is they got married. They have 2 children. They're still married to this day. Her name is Elizabeth Lewis, his wife, Liz. So married finally for Watson. Later years, 1990, among other things, he became the head of the Human Genome Project in NIH and did that for 2 years very successfully, and then got into a battle with the director at NIH because the director wanted to pursue the idea of patenting human genes, and Watson was very much opposed to that. I think his judgment was good in this regard, and he ended up resigning from that position. But sort of the kind of biggest controversy of Watson's career is that so in 1968 he wrote this memoir called The Double Helix that described his view of what happened in Cambridge. And the book had a huge impact. It was a New York Times bestseller. And I must say, when I started teaching biology in the early 80s, I would ask my big biology class, 300 students, how many people have read The Double Helix? And maybe half the students had read it. I myself read it when it first came out in 1968. I was digging it out here, and it actually was a birthday present to me in 1968. And I just loved the book. I thought it was fantastic. Now, the book had 2 big effects. One, it got people very interested and exciting about this new molecular biology and the way science was done. But there was also a strong reaction to the way he dealt with Rosalind Franklin in the book. Now, because he was very, very demeaning to her. And Crick was given a copy of the book early on, and other people involved. And Crick hated the book. And many other people strongly opposed the publication of the book, to the extent that at one point it was going to be published by Harvard, and they basically blocked the publication at Harvard. Watson got a European publisher who did publish the book, and as I said, it went on to become a real success. Now, Watson defends in his memoir, written when he was 70, that he wanted to write the book as he saw things as a 23-year-old, at the time. And so, well, in working for this course, I've got to go back and reread The Double Helix. And I must say, rereading it, it's still a great read, but I had forgotten just how many unkind, silly things he said about Rosalind Franklin in this book. So it's kind of curious to me that he became good friends with her, very supportive of her. And yet it's kind of a mixed feeling about the book, in that it was bad of him to say all those nasty things, especially since he wrote it when he knew they weren't true. But B, it was kind of bold of him to say, I'm going to write an honest book. This is the adolescent idiot that I was. And that's what he did. All right. So as I read through the book, I did get a different impression reading it now as an older person. What's interesting is that I think, having read all of this, I think Rosalind did not feel that she was overlooked. She left DNA very much her decision. She did what she wanted to do. She was recognized as an accomplished scientist. And in a real sense, Watson's book just kind of framed our historical view of that for many years. In a way, it's fading now, because now when I ask a class how many have read The Double Helix, the answer is always 0. Nobody knows this book anymore. So in a real sense, Watson's book brought Franklin more fame than she might have had without it, because of the reaction to it. But in reading the book, and I'm going to end here, and then I'm going to show you just a few pictures. I want to read the epilogue of the book. I hadn't remembered this at all. So as I told you, it's all exciting, all the, talks with Crick and the celebration at the Eagle and all about Rosie and how blind she was to her whole data. So in the epilogue, he talks about all the people involved and say what they're doing now. He wrote this, 8 years or so after the discovery. But then he gets to the end of the epilogue and he says, all of these other people that I've talked about can indicate events and details if they remember them differently. They can tell you now. But there's one unfortunate exception. In 1958, Rosalind Franklin died at the early age of 37. Since my initial impressions of her, both scientific and personal, as recorded in the early pages of this book, were often wrong, I want to say something here about her achievements. The X-ray work she did at King's is increasingly regarded as superb. The sorting out of the A and B forms of DNA would have made her reputation even better, was her 1952 demonstration that the phosphate groups must be in the outside, etc. Later, when she moved to Bernal's lab, she took up work on tobacco mosaic virus and quickly extended our quantitative ideas about helical construction into precise picture. Because I was teaching in the States, I did not see her as often as Francis did, to whom she frequently came for advice, or when she had done something very pretty. To be sure, he agreed with her reasoning. By then, all traces of our early bickering were forgotten, and we both came to appreciate greatly her personal honesty and generosity, realizing years too late the struggles that the intelligent woman faces to be accepted by a scientific world, which often regards women as mere diversions from serious thinking. Rosalind's exemplary courage and integrity were apparent to all. When knowing she was mortally ill, she did not complain, but continued working on a high level until a few weeks before her death. So Watson is just a mystery. I mean, why did he write the book that way when he knew that it was wrong? It's a puzzle. It's one of the puzzles of our time. So let me just finish by showing you just a few pictures. Okay. So here's Watson in his kind of second year as professor at Harvard, showing the model of the double helix. Here he is winning the Nobel Prize. This is Watson here, and this is Crick, and this is Wilkins. And the literature award that year was John Steinbeck, and that's Steinbeck here. Here is Liz. Watson now almost 40, finally love at last, and it seemed to work out very well in the end, I'm glad to say. So they did a story on this in this journal, this scientific research journal. Here is Watson as an older man, and here he is also in his office in Cold Spring Harbor with a model of the double helix in the background. And Watson and Crick as older men. Crick now ensconced at La Jolla at the Salk Institute, and Crick on the other side of the country at Cold Spring Harbor, both looking out over the water. And lastly, what became of Rosalind Franklin's chance for fame? Maybe one of the most interesting things is a news story recently that the European Space Agency has decided to name their Mars rover, which they want to land on Mars next year. They're naming it the Rosalind Franklin. So in the end, Rosalind has gotten kind of fame far more than, I don't know, maybe she would have wanted. And it's kind of an interesting view just of how the whole history of an event changes with time. And this whole idea of the controversy, is even kind of going away now as the whole influence of Watson's double helix on the reading of history is fading away. So that's my story. I hope you enjoyed it. I think three fascinating people I love the books. The books are still around, and I think reading probably the Double Helix is good, and I think reading the Rosalind Franklin book. I mean, those would be the two ones that I would really encourage. All right, questions. Let's see. Carol, you have a question? You got to unmute yourself.
Okay, I wanted to thank you. I thought the class was marvelous. I thoroughly enjoyed every single one of them, Barry. It was terrific. And I have a request for you.
Yeah.
I would like to know if you would consider doing in the future a CRISPR course.
A course what? On CRISPR, you said?
Yes, in the same manner in which you did this class, which was terrific.
Well, interesting way to do it is, as many of you may know, there's a new book by Walter Isaacson. He's written a kind of biography of Jennifer Doudna. Well, I like the idea. I haven't read the book yet. Yeah,
Okay.
maybe we can do that, and we could probably, maybe bring in a few other people too. Yeah. Okay. Good. Ron.
There you go. I have two questions and a comment. The first one goes back all the way to Rosalind Franklin. You mentioned that she was interested in working at, I think it was in 1954, in working on polio.
Mm-hmm.
And you implied there was a problem with it?
No, it later on, and I don't know how much further she got with it. Her specialty was really showing the detailed structure of the virus, but I know nothing more that it was just kind of illustrating that she was kind of broadening her horizon from the plant viruses to starting to work with animal viruses. But she died, rather shortly after that.
Right. Second question, you mentioned how Francis Crick would read for 2 hours to 8 hours. And I was just wondering, when he did that, did he just read, or did he mark up the books that he read, or did he take notes when he read? Do we know?
I don't know the answer to that. It's a good question. They have saved all of his stuff. They had many file cabinets worth of papers. So the answer is out there, but I don't know.
Oh, okay.
Yeah.
And then my comment is, it's wonderful to see that Rosalind's name has gone further in this galaxy than any of the other two.
True. Yep.
Thank you.
Okay.
Great course.
Thank you. Okay. Lois. Oh, you're muted, Lois.
Okay. The first is rather not very important, but I think it's interesting that Rosalind is always called Rosalind. Well, the two guys are always called by their last names. Mm-hmm. Some people have heard my theory that when people talk about their doctors, it's a woman doctor, they use the first name. If it's a man doctor, they say Dr. So-and-so. It's, I think it's a feminist issue. The second thing is, when I read the book, when it first came out, and I think everybody was reading it, mm-hmm. I was most impressed by the competition that scientists had then. Am I wrong in remembering it that way? That it demonstrated the competition between different labs, or?
Oh, definitely. I mean, Pauling was considered, he was the guy they all wanted to beat. And they knew there was a big discovery to be made. And that's certainly true today. Today, I think there's an extra element that's not a good one, is that you're competing for fame, but you're also competing for money. Yeah, and it was somewhat less so in those days, but the competitive element was really there. Yeah. All right.
I've got a question.
Nancy, yeah.
Or a comment. I thought it was interesting that when Rosalind was diagnosed and had that large growth removed. You didn't mention where its location, but that she very shortly thereafter died. I thought it would sound almost suddenly, of the ovarian cancer. And so I thought it was interesting because one might have thought that with the removal, they did some testing and that they might have also been able to have come across some of the early signs. And I'm relating this partly because I had the experience at almost the exact same time of having a large growth removed, and that they did check for the other, my anatomy to make sure that it was okay. So I just thought that was very curious. And here she is in the field, and I wondered if she wasn't really fully evaluated.
Yeah, and I don't know the details of that enough to really answer the question.
Yeah.
The implication has always been that they were connected, that they removed part of the growth, but that they were just subsequent to
I see. Okay. Well, that wasn't when you spoke of it, you hadn't mentioned that. So maybe they just didn't get it all. And maybe
And it was about, I think, 2 years between the kind of first one.
Okay.
She died.
Okay.
Florence, you had a question? And you're muted, Florence.
What you mentioned that Crick was schizophrenic.
No, one of Watson's sons has schizophrenia.
Oh, I thought it was.
No, it's one of Watson's sons. And I didn't mention today, in his later years, Watson became kind of controversial, to say the least, because he waded into this whole question of race and intelligence, et cetera. And he's never been a very politically astute guy. And so he has said some things that have gotten him attacked. Whether, he's really a racist or not, it's hard to know, I guess is the answer. But it's really tainted his reputation somewhat. Rusty has a question.
I'd just like to point out, I think it might be a little early to do a course on CRISPR, because I think what really makes this interesting is the historical perspective. And I thought you did a great job on that, Barry.
Yeah, thank you.
I agree. You made them come alive.
We all agree.
Okay, well, good.
Thank you.
I enjoyed doing it. It was very different kind of thing to put together. Yeah, but I've always enjoyed reading this biography, so it gave me a chance, and I really enjoyed going back and looking at them again. Okay, other questions here? I guess we're running a little over. Okay, well, thank you, and we'll think about. I mean, the CRISPR idea is interesting, and there are multiple people. Jacob and Manoa have really interesting stories, and there are some others. I also found there's a biography of Sydneybrenner I would like to read.
Yeah,
he's a really interesting guy and did some fascinating work. Of the players here, he's the one I actually met. Brenner one time. There was a big. UCSC was involved in the early days of the Human Genome Project, and there was a big conference on campus here, and Brenner came, and I met him, and I must say I was totally intimidated. I kind of shook his hand and hoped he didn't have any questions. Okay, well, thank you, everybody, and so hang in there with Oli. We're hoping to have some kind of summer activity. We're certainly going to start up courses in the fall, and we will all stay in touch. All right.
send in your renewal.
Yeah, send in your renewal. Renew your membership. Okay, thank you. Bye.
Bye.